Have you ever wondered why you look so similar yet so different from your parents? The answer is meiosis.
Meiosis occurs within the cell cycle in order to form you. Unlike in mitosis, which creates identical copies of cells, meiosis produces genetic variation among populations. This is a form of sexual reproduction which occurs in the cell membrane. Because of this, you're not an identical clone to your parents, but a genetic mix of both.
Before meiosis occurs, the cell must undergo a series of steps to prepare for division. This series of steps is known as interphase. The first step is G1. This is where the cell grows, develops, and differentiates itself from surrounding cells.
Following this step, the S phase occurs. This is where DNA is copied for each homologue and synthesized. The copied strands of DNA then attach to their sister chromatids.
The final step is G2 phase, where the cell prepares for division. It produces all the proteins it needs to survive as well as duplicates its organelles. Now the cell is ready to undergo meiosis.
First off, the cell has to go through prophase 1. This is where the chromatin condenses and eventually becomes visible chromosomes inside the nucleus. Once the chromatin condenses, a process called synapsis will occur where the newly formed chromosomes pair homologously.
Afterwards, the chromatids exchange pieces of maternal and paternal pieces with one another in a process called crossing over.
As you can see with these arrows, each sister chromatid will contain maternal and paternal pieces after crossing over. The nuclear envelope breaks down.
Next, the centrosomes and organelles specific to the cell cycle start to move towards poles of the cells. Each centrosome is made up of two centrioles, which are involved in the development of spindle fibers. We'll talk about those later.
The mitotic spindle starts to form and attaches the chromosomes at the centromere, which is the center of the chromosome. Here, the homologous chromosomes will stick together in pairs of four sister chromatids called a tetrad.
The tetrads begin moving towards the metaphase plate and line up. At this point, the nuclear membrane is completely gone.
Next step is metaphase 1. Here, tetrads will begin to move towards the metaphase plate, which is also known as the equator, and they eventually line up in a single file. Here, the spindle fibers, which are also known as microtubules, extend from the centrioles to create the spindle apparatus.
What's the point of the spindle fibers, you may ask? Well, these fibers attach to the kinetochore. You could think of the kinetochore as a belt loop on the centromere.
And we're on to anaphase 1, where each homologous pair is attached to a microtubule to be pushed and pulled to the opposite ends of the cell as the cell elongates.
In telophase 1, spindle apparatus goes away. Simultaneously, cytokinesis splits the cytoplasm.
A division occurs along the midline of the cell to separate it into two. In animal cells, this will form something known as the cleavage furrow, which is an apparent groove at the surface of the cell.
The nuclear envelope and nuclear membrane reform and reappear.
We're not done yet, so we have to go through meiosis 2 in order to get our end goal of 4 daughter cells.
In prophase 2, the nuclear envelope breaks down once more and the chromosomes condense again. Just as before, the centrosomes start to move towards opposite poles of the cell.
By now, the spindle apparatus is formed. Next, we arrive at metaphase 2, where once again the chromosomes line up along the metaphase plate, but this time it's the individual chromosomes that line up and not the homologous pairs. Aligning at the midline, this is known as independent assortment. Just as before, microtubules attach to each chromosome at the kinetochore of the centromere of the chromosome.
Afterwards, anaphase 2 takes place where each chromosome is split in half where the sister chromatids are attached to the microtubules to be further separated. Now, sister chromatids of each chromosome separate and start to move apart to opposite poles of the cell. Each sister chromatid has now become its own separate chromosome.
Finally, we've reached telophase 2, the final stage of meiosis 2. Similarly to telophase 1, the cytoplasm will divide again through cytokinesis in telophase 2. This time, cytokinesis will form two cleavage furrows. The nuclear envelope and the nucleus of each cell reforms and the spindle disappears once more. We have now reached our end goal of 4 cells with similar genetic variations. Remember, meiosis is what makes gametes for sexual reproduction. In males, 4 sperm cells are produced after each round of meiosis. In females, one fertilized egg is produced as well as 3 polar bodies, which contain the same amount of DNA, but is much smaller than the egg cells, and they don't have a cytoplasm or organelles.
All in all, without meiosis, you would not be here.
I hope you learned something new and that this video was helpful. Until next time.